We are here today with Professor Marc Rothenberg, Director of the Division of Allergy and Immunology at the Centre of Eosinophilic Diseases and the Principal Investigator at the Consortium of Eosinophilic Gastrointestinal Research, normally known as CEGIR. We're very excited because he's published over 500 papers, no mean feat, and has made significant contributions to the eosinophilic disease community. And in the advancing and understanding of these complex conditions. I've had the pleasure of knowing Marc for over 20 years, which probably makes us all feel old, and I have been working with him for the last 6 years within CEGIR, and are really proud to be doing that. I personally have witnessed significant progress in this area from the early days when I first met Marc, when he was basically didn't even have a big building to work in. And so the meeting that we had was in a hotel downtown, to where they are now with the big research centre that is available in Cincinnati. So welcome, Marc. Welcome to EOS Network, and thank you for joining us for this podcast.
Good day, Amanda. It's a pleasure to be here, and thank you for the kind introduction. And it's been great to partner with you in this journey to understand and treat eosinophilic diseases.
Thank you. I think I would like to start out with, first of all, a question that probably a lot of people would like to know the answer to. So what inspired you back in the day as a medical researcher to actually get into the field of medicine? And also, how and when did your journey begin with the eosinophil?
Wow, Amanda. If you want to go back to the beginning, my interest in medicine probably started in the cradle. I was just a curious kid, you know, and my parents would say when we would go places, I would make observations and I'd ask them to explain things that other people wouldn't notice. I thought that understanding things was something that inspired me when I was young in high school. I participated in different scientific inquiries through testing different things that were well known already, but I wanted to prove them to myself, like how did a camera work? I became interested in chemistry. I wanted to understand how film was developed and set up a chemistry lab in my bathroom. My parents also let us build a dark room in another bathroom. So there was a lot of curiosity. I was inspired by my parents, really, and my mother, who has a chronic medical disease of childhood diabetes and is insulin dependent, and she's taken well over 100,000 injections in her life to keep her alive. And I became aware that if she missed 1 or 2 doses of that medicine that wasn't available 50 years before she got diabetes, that she wouldn't have been able to survive. So it inspired me to know the power of medicine and the power of research, and to have my own quest to understand the way things work, and eventually to focus on our medical issues. When I pursued college, I was undergrad with a major in biochemistry and chemistry, and the reason I majored in that was because I sought out the leading researcher at Brandeis University, a world-renowned biochemist who was a great mentor, late Professor William Jenks, and I was always in search of excellence. That I always wanted to align myself with the greatest scientific minds. And what I learned from Dr. Jenks and all my other great mentors was how little they knew, and even though they were the world experts in what they worked on. Jenks taught me how to ask fundamental questions about simple processes. And I subsequently went to medical school, MD, PhD program. I wanted to pursue medical research, but I wanted to make sure that I could understand medical problems from the point of view of diseases, and got an MD in addition to a PhD in immunology. My interest in immunology was focused in on understanding how the body protects itself, how it repairs itself. But again, similar to college, I sought out the best mentors I could find. I was stimulated by a professor, the late K. Frank Austin, who was one of the leading inflammatory researches in immunology back in the day, and is still listed as one of the top biggest contributors to immunology in the last several decades. And he just passed away. And I was inspired by him and came to him, and he attracted me to the field of allergy, which he was an expert in. My interest in eosinophils was serendipitous because I was working on a related cell, mast cells and basophils, and ran an experiment on basophils, but used the eosinophils as a control. And the eosinophils turned out to not behave as they were expected. The basophils weren't as even interesting as the unexpected result that we got with the control, which was the eosinophil, what we call eosinophil survival, which we discovered at that time was against the current paradigm that eosinophils were short-lived cells from the blood, but we found out that they actually underwent maturation and what we call post-mitotic differentiation outside of the blood, which was completely unexpected, and these results led to the subsequent discovery of the eosinophil poietins like IL-5 and GM-CSF and IL-3, and then the 20 years of subsequent work, which I contributed to in part, which was the development of the antibodies and proving that anti-IL-5 and eosinophil therapy was very useful for various diseases in patients, and of course the FDA approval of the new class of medications, which was based on this work. After the PhD, and maybe this is a long answer, Amanda, you can say if I need to shorten it, but I pursued pediatric training. I always wanted to help people, and I thought kids were the right point of entry, get kids, get a disease process during its development. And pediatrics was also, for me, not as depressing as taking care of aging and deteriorating and degenerating adults, but rather take care of young people as they were developing lives ahead of them, and the idea of intervening early in a disease and potentially reversing it, especially with the immune system, which we know is reversible disease because of the ability to modify memory responses and tolerize to antigens. During my training, which I did in pediatrics, then subsequently in allergy, immunology, and hematology and oncology, a lot of work, a lot of different types of amazing training and great mentors along the way. I ended up doing my postdoctorate research in the Department of Genetics at Harvard, where I was already training for the prior decade, and seeked out the best I could find, which was the Department of Genetics, the chairman of genetics, the late Dr. Phil Leader, who was also a pioneer in the field and actually was participant of deciphering the genetic code in terms of the triple codon theory for understanding how the genome is translated into proteins. So Phil Leader was an amazing mentor and scientist and had a peripheral interest in allergies, but I figured I would work with him because I would learn another area of science, which was genetics. And we, in his lab, along with my colleagues, discovered and molecularly cloned the eotaxin family of chemokines, continuing to pursue the eosinophil interest that I had, but also extending it to a different pathway that was unique, but cooperative with IL-5, which was the understand how eosinophils accumulate in the tissue after they develop, and the eotaxin family of proteins, which regulates eosinophil activation, and chemotaxis, which is the movement of the cell, and subsequent understanding of the life cycle of the eosinophil through that work. And a number of things fell out of that. And then the recruitment to Cincinnati Children's to work on allergy, an amazing place here at Cincinnati Children's, the first research foundation formed in the U.S. in 1931 in pediatrics, and the development of amazing clinical and research enterprises you've seen here, Amanda, when you visited and participated in some of our meetings. But what I really love about here is the team that we've built. Great people that are interested in this disease, eosinophilic esophagitis and related eosinophil GI diseases, which we helped to put on the map and described it initially, but then developed the animal models for the disease, and then subsequently shown that it was related to allergy rather than intrinsic problems with the immune system, but more related to external problems where the allergen, particularly food allergen, was triggering these responses, and deciphering the mechanism related to the allergic arm, discovering of the specific mediators of that, which provided the framework for the first FDA-approved precision therapy in these diseases, as you know, the antibody against the IL-4 receptor, which is dupilumab. So long journey. It's been lucky and blessed to have great opportunities to be inspired by patients like you and your group, Amanda, but so many others, especially the Cured Foundation that's been supporting our research and leveraging our ability to use federal funds here in the United States by the National Institute of Health, along with great colleagues, particularly associated with the Consortium of Eosinophilic Disease researchers, which is really an amazing group of people with different backgrounds and different interests and amazing skills coming together with patients and the NIH to solve the mysteries of these diseases and to advance the treatment. And still, my goal is to cure the diseases, which is an ongoing quest in the laboratory.
Absolutely. And I know that we could keep you online forever talking about the work that's already undertaken. Do you think within your joint journey, are there, I mean, I know you've talked about the IL-5 and the IL-4, and to people outside of the research world, they won't necessarily understand what that is or the process and how long it takes. I know personally that when I first met you in 2004, those research was actually in its fledgling field, just beginning then, and it's taken all this time now to get to the point of actually having a drug. And I think that's something outside of the medical world is difficult for people to understand. Are there some real key moments, do you think, in that couple of decades that you think have got us to the pivotal point that we are now?
Yeah, there have been a number of key pivotal moments, and I like to think about all the unexpected results we had. I always tell people in my lab, you can work on anything you want as long as you produce results rapidly in papers. And the best results are ones that prove my theories wrong. So I love finding unexpected results. And there's been about 10 occasions in my career when we had unexpected results. I described one, which was that eosinophils were not expected to be long-lived cells. We had a number of other observations, but I think, for the sake of focusing today, one of the most unexpected observations was one that we just reported, that despite 20 or more years of research and development of eosinophilic esophagitis as a disease called eosinophilic esophagitis because it's an eosinophil-associated disease. Eosinophils are really a selective accumulation of these in this disease. And it's used to enumerate, you need to enumerate the cells. It's established and agreed upon internationally. The eosinophil levels should be X, Y, and Z. We can go into what they are, and they should have certain characteristics because this is an eosinophil-associated disease. And indeed, we showed that in experimental systems in a number of ways. So we finally created in humans through a very safe approach, so I like to call the eosinophil human knockout, which is something we did in mice, but now we can do in patients, which is use safe drugs which have been developed to eliminate eosinophils in the body. And we definitely predicted, along with many others, including the companies that were developing these drugs, that this would have a huge impact on helping patients. And after a long period of research, of course, early stage phase 1, phase 2, and then phase 3, which is studies that involve many sites in many countries, many patients that have decided to be involved in research and going on a drug or a placebo, which is a very substantial undertaking by all the involved parties. We were very excited to do this experiment in humans, but when we finished the experiment after all this work, we found out, and lo and behold, as we planned, the eosinophils were removed from the patients almost entirely. But the patients unexpectedly did not show clinical improvement. And we did report those results recently in the leading journal here in the United States, the New England Journal of Medicine, which is, really the top of the line journal. And the results are really transforming the field, because what they're saying is that even though this is called an eosinophilic esophagitis, and even though eosinophils go up to unbelievable levels, and we use eosinophils to monitor the disease, to diagnose the disease, these are not the primary driver of the disease. So that's an aha moment. Coming back to your question, that's a real pinnacle in the field. Unexpected results, and it greatly impacts patients. and it's impacting the field because it's changing the way we think. It's giving us pause in terms of reconsidering how to diagnose, potentially monitor the disease, and of course, how to develop future therapies that aren't necessarily focused on lowering eosinophils, but also not necessarily monitoring or testing the ability of various therapeutic interventions only based on the eosinophil levels, which is often what we've done in the past and what the FDA and the other regulators have indicated is very important. So we are reconsidering this and pushing forward with a strong agenda done to understand this and then to see how we could potentially use this new information to help patients.
Thank you. Thanks, Mark. I think, absolutely, this is a key moment. And it's interesting, you thinking about almost feels like we've done full circle. We're now talking about eosinophilic associated diseases, which I think when our knowledge was very small at the beginning, that was kind of a term that was used quite a lot. Then it kind of disappeared for a period of time, and now the picture seems to be opening up much more. And it's almost probably feels a bit like you've done full circle with your research since you started out with mast cells and basophils and eosinophils, and we're almost back to where you started. Thinking about the benzelimab trial, and we will provide for the future of anybody who's listening to the podcast or looking at this video, we'll provide the link to details of that on there. And we're actually really grateful that the data from this, despite being you could consider negative for the Pacific trial, was actually published, because as you say, the negative data was just as important at moving forward for future research as actually positive data. So we're very grateful for that to have been released. So thinking about where we are now, what do you think the next 5 to 10 years of focus is going to be? And if you hypothesized, where do you think this, and how do you think this will change the patient's experience of living with the eosinophilic associated disease in the future?
Always good questions from you, Amanda. Just to step back one bit, like you said, we managed to publish this data, which is important because it is negative data. But we recognize that even though this was negative data, that this was going to be a landmark information. And we took the bet and sent it to the leading journal in the field. And indeed, they agreed with us and were very enthusiastic about also releasing this information. So it shows, and I encourage, people to feel good about participating in research. Everything does advance the field. Even if the results don't turn out to be what's expected. It's important for people to report negative data. It's important for companies to do that as well. And I commend the sponsor for being behind that, because that doesn't always happen. The sponsor of that particular study, which you can read about. In terms of where the field's moving, there's certainly now an exciting amount of what we call precision therapies being developed, which is the ability of drugs like dupilumab to block different components of the immune system and to do it in a relatively safe manner. Your patients, your members of your organization, those interested, and importantly, those suffering from eosinophilic GI diseases, should know that what our research has shown is that the allergic arm of the immune system is primarily vestigial. And what I mean by that is it's not a required arm for daily health in most people. So unlike other components of the immune system with other drugs that people are on, and I can go over which ones, there's often immunosuppression. What we see with the allergic arm is that we can actually eliminate it. We can eliminate eosinophils. We can knock off a major pathway like that blocked by the dupilumab. And patients generally are fine. That, when we can talk, when I say fine, they're basically okay. Every drug has its side effects. So this is encouraging for the future, because there are a number of other ways of interfering, blocking in safe manners with the allergic arm of the immune system. And there's a series of new targets and drugs at all stages of development, including late stages, that are being tested to be, and I predict will be very impactful for eosinophilic esophagitis and related diseases of the GI tract, the EGIDs. In my own research, we are heavily invested in that, developing the proof of concept many times for these drugs, for these therapeutic intervention strategies. But we're also trying to think big. because the main goal ultimately, when we can discuss this, I think for patients is to not have to take medications. Diet is certainly something that's close to maybe more natural, and we know that it can be very effective. And we have good data now, especially the recent studies by the CEGIR group, which are the first randomized multi-site studies on diet. But we want to do more than just that. And I'm encouraged by the fact that some of the greatest breakthroughs in the last in this century, medical research has to do with the immune system, particularly the ability of the immune system to cure cancer by manipulating the immune system with immunotherapy, and also the ability of the COVID pandemic to really be greatly impacted by a new technology, the mRNA vaccine technology. All of these are basically working by modifying the immune system. So my predictions, Amanda, are that we're going to see a huge amount of application of this research. The immunotherapy, the mRNA vaccines, both of those things we're heavily engaged in now to really have impact that I hope will reverse eosinophilic GI disease, allow patients to assume normal lives without taking chronic medications, and to re-educate the immune system and hopefully cure the disease through this type of medical research.
Thank you, Mark. And that kind of leads me nicely into a topic which I think is quite complicated for everybody, both patients and healthcare professionals, is understanding now as this field evolves, the association with other conditions, the term associated eosinophilic disease and comorbid diseases, and how that all interacts together. And I wonder if you could give us a little overview of that and why that's important in research and treatment development, how you think that will play a part.
So what you're asking about is the association of EOE and other eosinophilic GI diseases with other comorbidities. Is that right?
Yeah, correct. So obviously we're seeing an involvement in these treatments that are actually being multi-purpose now for both for skin, for asthma, for nasal polyps, all often allergic type diseases, and that actually finding the same treatments are working. But also there is the complicated topic that sometimes these can be a specifically associated disease, or they can be a comorbid disease dependent on their severity. And it'd be great if you could sort of explain that a little bit more beyond my terminology. Would be really helpful.
Well, we've discovered over the years, and this is also one of the unexpected findings. When we first started working on EOE, if you were to ask nearly everybody what was the disease caused by, they would have said acid reflux. And part of that reason was because that was the main disease of the esophagus. Reflux is a common disease. And also anti-reflux medications like proton pump inhibitors would use to actually treat EOE. We subsequently showed in animal models, we could talk about what they look like, that we can elicit EOE, not with acid, but with allergen exposure. And that led to the idea that EOE was an allergic esophagitis. And the eosinophils were part of the allergic response, and it was driven by food allergens and the immune hypersensitivity to food. So the reason this is important is because typically EOE does not occur in isolation. It can often, and it typically occurs in nearly everyone, in an individual that's an allergic individual. We call it an atopic individual. Atopic means that you have one of the allergic diseases, which whether it be eczema, asthma, allergic rhinitis, which is typically hay fever, or it could be food allergy, for example. And we now understand that allergies are a spectrum, and there's often transition from one to another, and that can happen in an individual. It can coexist in an individual, or it can happen on a population level. We can see that the age of onset of some of these diseases is different, and there's something we call the atopic march, which is a very simplified way to describe the association. And we now understand that EOE is part of that. So most patients with EOE do have comorbid allergic disease. typically the EOE is actually the more of a substantial problem. We don't understand why, but in most patients, most of the other allergic diseases are not severe, but they're there. They can wax and wane, while the EOE is more chronic and can be the highest morbidity amongst the other atopic diseases. But that's not true for all patients. Some patients can have a whole body that's inflamed and sensitive, and they can have eosinophils in the blood, and they can have severe skin problems. Those patients are often part of more of a syndrome. Some of that is linked to different genetic causes, can run in families as well, and we've pinpointed the genetic basis of that, as well as the common presentation for EOE, which is related to genetic susceptibility areas in the genome that have been very informative. So EOE is part of the atopic march, and it's atopic disease, and the comorbidities can be related to that. But also, Amanda, over the years, we've discovered that EOE can be associated with other problems as well. It's not uncommon for people to have other inflammatory diseases, especially an enrichment of inflammatory bowel disease, as well as celiac disease, and another type of presentation, which is a hypermobility, which is meaning the joint laxity and some of the problems associated with that could even including fainting and we call POTS disease, which is um, blood pressure instability with position of the body, postural orthostatic hypotension, which is POTS. In any event, there are a number of morbidities, and having a chronic disease like EOE or other agents is tough. It's a tough challenge, and it can lead to hardships in terms of people's behavior and psychological profiles, and it is tough. But I just want to say that I'm just so impressed with people that have these diseases and what they've achieved. I've seen people at all ages excel. I've seen some leaders compared to their peers. This disease can often be motivating because it's a treatable disease. It's one that you can live with. Patient support groups like the Eosinophilic Network is so important to bring people together and inspire them and to provide support at all different levels. So, although there is a lot of comorbidities and the disease is tough, it's one that can be used as a challenge to achieve high levels like you've done with your family, Amanda.
Thank you. And I think I've met some very impressive young people, I think, at some of the conferences. And I think last year we met even a researcher who's now taken the journey through medical to try and research the disease himself, no more inspired than by living with the condition. So definitely you're seeing people fight their way through it. And it is challenging, and we do see many people, and I think the mental health impact of living with this disease is often not mentioned, but actually we do see it every day. And I think there's two sides to that. I think there's two sides, that the mental health impact is not just from living with the condition, but often from the challenge of having to get the right medical care. We know internationally it's still a challenge across the world to get the right care and the right treatment, and we continue to advocate for people for that. I think one other question I would like to ask, we've got a couple of questions. We did ask our community whether there's some questions they would like to ask you. I mean, you can imagine there's hundreds, so we've kind of try and summarize them for you. There was a lot of questions around future ways of testing and looking for different biomarkers. I wonder if you could first explain what a biomarker is. I think that would be very helpful for our community, and give us an idea of some of the trials that are going on around the world, are ways that we will be looking to test and manage and monitor the disease going into the future.
So this is a really good question. The idea is that right now, the gold standard for diagnosing and monitoring eosinophilic GI disease is through an endoscopy. An endoscopy is by itself, not a straightforward procedure, although it's readily done and it is pretty safe. But it's kind of annoying to have to do. I mean, to go to the doctor to get a tube put down your throat, to be sedated, and kids to have to go in anesthesia, the risks of anesthesia, and the unfortunate need to recurrently do these procedures to monitor the patients. Because there's currently no acceptable, what you might call, marker for the disease outside of the endoscopy, and then looking under the microscope at the biopsies, which is a costly procedure. It's time consuming, and it's what you call invasive. So there's been a quest by my lab and probably almost all researchers in the area to develop other ways of diagnosing and monitoring the disease. And we are heavily invested in developing a blood test for EOE. And we have made progress with that. And we're seeing exciting results now, which are better than ever before, related to not only the genetics, but also to particularly looking at molecules and proteins in the blood that we think have great potential to be that test that we've been looking for, a blood test. And what we call these markers that aren't exactly the gold standard right now, which is the biopsy, we would call them biomarkers. Biomarkers are things that are biological and they mark a disease or a phenotype. In this case, we're talking about the best case scenario would be a blood test that we can use to diagnose and monitor disease. And an example of that would be a test that we can use to diagnose, for example, and monitor prostate enlargement in men. That's something called a prostate-specific antigen. My lab is looking for what we call an eosinophil-specific antigen, an eosinophilic esophagitis-specific antigen. And progress is being made in that regard on this test, and we're excited about it. Hope to share that with you in the near future. But there are a number of other ways that are being developed. There's a number of different types of molecules that people are looking at. These could include other molecules in the blood, like microRNAs, which are different molecules. We wouldn't necessarily go into that, but there's also now, one of the things that happens in research is that there are certain fields that lead and pave the way, and I think cancer leads the way, and what we do in my lab is to keep on the cutting edge of the breakthroughs that are taking place in different fields and to apply that rapidly to the allergy problem with a strong focus on EOE. So one of the things you've seen, and you've probably maybe, a lot of your members may even be thinking about getting is, and they have blood tests now for cancer. And there's something called liquid biopsies, which is an idea that you can actually take a blood test and get biopsies of different tissues. So we're obviously very, very interested in liquid biopsies for the esophagus. And there's a lot of research going on in that area by many different groups. Besides these tests, which we would consider to be really non-invasive tests, there's things that are called less invasive tests, which is basically things that are available now. And one of them is a string test called the eosinophil string test, which you basically swallow a string and it stays in there for a certain amount of time. Then you pull it out and then it goes to a lab and they measure different proteins that they take off the string. And that's an example of a non-invasive test that's got a lot of potential and is being rolled out now. There are other things that are being developed that are available. One is instead of going down with a regular tube down the throat and doing a full endoscopy, there's a process now called transnasal endoscopy that's basically allows a patient to be awake and to actually pass a small tube, an endoscope in the nose, and then to pass down the mouth in a conscious, awake individual, including children. And that can also be used to call less invasive monitoring of the esophagus. And the possibility that could also be used for below the esophagus is being developed. So there's a number of other things going on. There's this group looking at imaging studies using radioactive, sounds pretty bad, but people do take safe, what you call radionucleotide labeled markers or traces, and you can swallow that and then potentially monitor inflammation in the esophagus. That's also being developed. What I think is exciting, Amanda, is that 10 years ago, none of this was available. And we talked a lot about it, and now we have options that are actually available. We don't yet have an agreed upon non-invasive blood test, but I think, if we come back here in 10 years and hopefully shorter, based on the data that we have, it might be much shorter than that, that we're going to be seeing really other transformative research impacting this particular question about how to monitor the patients and diagnose the disease.
Thank you. I think all over the world there's some amazing research that's going on. I know here in the UK there's poo sample testing. They're looking at that, urine. The also a cytosponge here, which has been approved for Barrett's esophagus and is being trialed currently for EOE, which looks really interesting as well. And for information for anybody who's listening to this, who's hearing a lot of terms that they don't understand around clinical trials, there will be a link at the bottom of here for you to be able to find it, but we have a module that will educate you now on the process of what a clinical trial is, and you'll be able to understand a lot of the terms that we're using, how long a trial takes, and have a real general understanding if you actually just want to understand the process, or if you're thinking of considering and taking part in a trial and you want to know, what does it mean? What does it mean if I'm signing up to this? What questions should I be asking? If you work your way through this module, which is available in written format or in video format, it should give you all the tools that you need. So talking about trials, I think we wouldn't be able to complete our podcast without giving us a moment to really give a massive shout out to the work that's being done at CEGIR. I would love you to tell us about CEGIR, how it began, and give the people who are listening to this an idea of what that actually looks like today, how many people are involved. I personally, one word springs to mind when I think of CEGIR, and that is collaboration. I am incredibly proud to be part of it. I believe that the way that science has moved forward in what outside probably doesn't seem fast, but actually has been incredibly fast for what's been achieved, is the fact that you are completely collaborative with everybody and anybody. The open data sets that are available via our website, that you encourage people, even outside of CEGIR, to use the work that you've achieved to further science. So if you could just give us an outline of how it began and who's involved, I think that would be great for people to hear.
CEGIR stands for the Consortium of Eosinophilic GI Disease Researchers, which started about 12 to 13 years ago. It took a couple of years to get funding, which was primarily by the NIH through a competitive process associated with the National Institute's Rare Disease Clinical Research Network. And CEGIR's goal is to understand, educate, and treat patients with eosinophilic GI diseases through, as you said, collaborative research. CEGIR is a partnership. It's a partnership with patients. It's a partnership with the key stakeholders in disease. And it's a cooperative agreement with the National Institute of Health, funded by 3 different institutes there, which is the NCATS, the National Center for Advanced Translational Research, the NIAID, which is the National Institute of Allergy and Infectious Disease, and the NIDDK, which is the National Institute of Diabetes and Kidney Disease, and GI digestive disease. So it's amazing to see the interest at the NIH level, because we've got 3 different institutes that are coming together. And it's not just a grant where they give you the money, but it's actually one where they partner with you. And NIAID is leading that partnership, so they're active participants in the process. And very central in the process is the partnership with the patient advocacy groups. The primary groups in the US is the CURE Foundation, which is the Campaign Urging Research for Eosinophilic Diseases, as well as the ABFID group, the American Partnership for Eosinophilic Diseases, as well as the Eosinophil Family Coalition, or EFC. And of course, we're very pleased to partner with you, Amanda, as another partner, with the EOS Network, and we have a group in Australia, the EOE, Aussie EOE. And then there's a number of other partners. But CEGIR primarily involves 18 sites in the US, 18 of the leading sites coming together to solve the mysteries of these diseases, as well as other partners in different regions of the world, including Europe. And then we do partner with other key stakeholders, including drug companies that are interested in this. And the funding does also come from some of these groups, particularly the patient advocacy groups, CURE and ABFID, as well as more recently, certainly some of the institutions that are involved, particularly Cincinnati Children's, which is a major supporter of this area of research, as well as Colorado Children's and the University of Colorado, and now some of the other institutions that are involved, the other 16 institutions are also supporting us in many different ways. Of course, they're supporting us by developing the great researchers and clinicians that they have at their institutions, which is too numerous right now to count, because there's really wonderful people at each of those institutions that are coming together. And that cumulatively is about almost 100 people working, every day to advance understanding and treatment. Part of CEGIR's mission is to educate the next generation of leaders in the field, and one of the core processes in CEGIR is a career advancement and educational core that it has identified already over 20 different people at early stages in their career, MDs, and are developing mentorship plans and allowing them, as you said, to benefit from the CEGIR data that's emerging. One of the studies in CEGIR is to follow patients over time, a longitudinal observational cohort, which is basically following about 1,000 people with different types of eosinophilic GI diseases over time. And we use that data to uncover natural history of the disease, but also to ask fundamental questions about how to diagnose, how to monitor, how do we build different tools to become standardized, which are very important for regulatory agencies to use to approve drugs in clinical trials, such as how to count eosinophils, which is not a trivial matter. And we do have a whole core of expert pathologists that are involved in that. One of the nice things and amazing things about CEGIR is that it's dynamic and it changes over time. So we just had a call earlier today, and we're talking about how to use the latest information to build new guidelines that are internationally recognized with international partners to better diagnose and treat, understand these diseases, and to do that in partnership with different key stakeholders, especially the regulatory agencies. And CEGIR is mainly involved in collaborative work with the FDA.
I'm very proud to say that you're working on currently, and I think there'll be many people who are listening to this to be keen to hear the work that's happening in lower gastrointestinal diseases. And it feels like we're a way along the way with EOE. I'm sure there's still much more to be learned with that, but actually now the work is really beginning to come forward in the more complex, less recognized lower gastrointestinal diseases, but it's coming, and I think that's something to be hopeful for the rest of the community and for the healthcare providers that need these guidance and need the tools to be able to do the job. So we're very appreciative of that. What I would say is I would encourage anybody who has the opportunity to work with CEGIR to grab it with both hands. They're very slick. I've never been in a meeting like it. Believe me, it starts on the dot, it ends on the dot, and they manage to get everything on the agenda in between, and it's well attended every time, hence why the research moves forward in the way it does. I would also encourage anybody who's listening to this who isn't registered with us as a healthcare provider or as a patient to do so. We do what we say on the tin. We connect people together, and we bring peer expertise together when it doesn't happen, and we find support for people who need it. So I encourage you to click the join button on our website if you haven't done so. Join the nearly 700 healthcare professionals that we've got registered with us now in around about 45 countries, I believe, and our patient and carer community. And I hope you've enjoyed our presentation and talk. I'm incredibly grateful for Marc giving up what is a very precious commodity in his life, which is time, to come along and speak to us today. And we can't wait to see what's next in the research that you're doing. Thank you, Marc.
Thank you, Amanda. It's my honor to do this, and it's why I'm busy is just because of things like this and being inspired and trying to partner with patients to make their lives better. And this is one step along that process. I want to give you a round of applause for what you're doing with your organization. Thank you very much to all your supporters and membership to advance the field and to call the attention to the right people in this important disease in an effort to improve those suffering from it.
Thank you so much.